Welcome to Office Hours on recapping Unit 3.1 through 3.4. It's all about the energy in AP Biology. So, way back in Unit 1, when we learned about macromolecules, we asked the question, why do we need to eat? And we said that in order to maintain our bodies, to repair and to grow, we need matter. We are made up of matter, so that's why we eat, to build up our body, as well as the plants need the carbon dioxide, water to make up their bodies through photosynthesis. Now, my question for that unit and our question will be, why do organisms need energy? So this is our topic. So we will answer that in a little bit. Let's divide the organisms into autotroph and heterotroph organisms. So we have, let's screen the autotrophs. Autotrophs are the ones that they can make their own food. We have the low energy molecules like carbon dioxide and water. We add a little bit of sunshine with its energy, high energy, and the plants are making bigger molecules than carbon dioxide and water. With high chemical potential energy in the bonds. What do these organisms use those big molecules? They are using it for growing, for repairing, making molecules, repairing cells that they are lost, as well as making ATP molecule, and one more, maintain homeostasis. Please, I want to point out, homeostasis is not equal to equilibrium. Equilibrium means equals on both sides. Homeostasis, a special level of statement inside of the cell, no matter what the outside is compared to the inside. Homeostasis is a state that is stable inside of the organism, in the cell, and an organism, the organ system. Now, let's see what is happening with the autotrophs. We check that and check the heterotrophs. The ones that they cannot make their organic material out of inorganic material. So heterotrophs, they are going to eat the bigger molecules with energy, or they eat each other, okay, the carnivores, for example, and the omnivores. So we have the bigger molecules with the high energy, potential chemical energy. So these heterotrophs are all also using this bigger molecules with high chemical potential energy. They are using it for growing, for repairing cells, for maintaining just making ATP, as well as maintain their homeostasis. So these are the common goals why we need energy in our system. While the matter recycles in the ecosystem, the energy is not going to be recycled, will be used and dissipates. So almost the ultimate source of energy is the sunlight in our planet. We will talk about a little bit later on the geothermal energy also, but that's really the ultimate source of energy. Okay, so we are done with why we eat and why we need energy from the food. The other one is we mentioned the ATP. So our topic, the subtopic will be the ATP. What is the use of the ATP? Why do I need ATP in my body? So let's say we have substrate A, and we have a substrate B. In order for the substrate to turn into substrate A to B, let's say this is a big molecule, a polymer with high energy. This will be a monomer going to be broken down. So this is an exergonic chemical reaction, losing energy along the way. Why do we need that? Well, we have ADP and inorganic phosphate molecule. In order for the inorganic phosphate molecule to add to the ADP for high energy molecule, well, it needs the energy from the exergonic reaction to turn into ATP. This is how ATP can form one way. This exergonic reaction fuels the ATP production. This is a coupled reaction. Besides that, we have one thing that is going to go out, water, from this exergonic reaction. And we try to use this, and it's going to get the water out. Good. Now, going back, this is low energy. This is high energy material. One more thing, all of the energy that in substrate A will not be converted to AP molecule for the making ATP molecule. Some of the energy leave the system in form of heat. That's important for the efficiency wise. The next one is going to be fuel, like we have substrate C and we have substrate D. I'll put this one a little bit down, substrate C. In order for, this is, let's say this is a monomer, less complex, and this is a polymer, more complex, have higher energy level, lower energy level. In order for substrate C to turn into substrate D, that is an endergonic reaction. Reaction requires energy input, and at this point, the ATP is coupled with that reaction and provides the energy for the substrate C to turn into D, fuels that chemical reaction. Now, compartmentalization. We discussed that in Unit 2 when we learned about the organelles. In eukaryotes, we have membrane-bound organelles. Why are they so important? They increase the efficiency of the chemical reactions, the different chemical reactions that they can happen at the same time in the cell in a different milieu, for example, to be the cell to be more effective. Many different chemical reactions can happen at the different times. That they require might be different pH levels inside of the organelles compared to the cytoplasm. Now, the third topic, what I would like to talk about, now we talked about the ATP. How do those reactions happen? Okay, the endergonic and exergonic, the reactions. So we will have two diagrams. This will be one of them. Let's say this is the energy level of the substrates and the products, and this is the progression of the chemical reaction. Let's talk about the exergonic reactions. When they release heat, basically, or we start with a substrate that's higher in energy level and the product is lower in energy level. Normally, the substrate needs to gain a certain amount of energy for the bonds to get loosened up, and after the reaction can happen. So this is the substrate, and this is the product. This is the activation energy. And to lower the activation energy, we have some enzymes. So with the help of enzymes, the activation energy doesn't have to be that high. It can be only that activation energy with enzymes. Enzymes are biological catalysts. They lower the activation energy for the chemical reaction to take place. In my body, the exergonic reaction is the cellular respiration glycolysis. I have the glucose and breaking it down, oxidizing it first to pyruvate and later on to carbon dioxide. I'm burning the sugar, but I'm not in fire at all because I have the enzymes that they lower the activation energy to facilitate the reaction at a lower temperature. Okay, this is the exergonic. Let's discuss the endergonic energy diagram or endergonic reactions. We have the energy level and the propagation of the reaction on the x-axis. We start with a substrate with low energy level, and the product will be at the higher energy level. Where would the product be at the energy level? Also from ATP, and more chemical potential energy will be stored in here. Endergonic reaction, for endergonic reaction, example is the photosynthesis. So the substrate will go up like this. This is the amount of activation that the substrate needs to gain to turn into product. If we have enzyme, the activation energy will be that much. It requires only that much with the enzyme. So the enzymes are super important, and Now, this leads us to another question. So what are enzymes basically? What are those? Many students like enzymes. Enzymes are fun to learn about, and they make most of the times sense. So we are going to talk about some enzymes. Enzymes are proteins, and we learned that proteins have primary, secondary, tertiary structures. Some of them, when two proteins, polypeptides, combine together, they have quaternary structures too. The primary structure comes from the sequence of amino acid, secondary if they have alpha helix or beta pleated sheets, and the tertiary structure comes from the interactions of the R groups of the amino acids. What interactions I'm talking about? Hydrogen bonds, disulfide bridge formation, cysteine and cysteine, amino acids. I'm talking about the positive and negative interactions, attractions between the R groups for some of the amino acids, as well as the hydrophobic interactions. The phobic parts gather around inside of the amino acid. They do not want to move towards the watery substance around the protein. So these are the interactions that make up the tertiary structure. So the enzymes, let's focus on two of them at this point. Let's say we have one enzyme and enzyme A, and we have a substrate in here. Let's make this to be an enzyme that facilitates endergonic reactions. So here is one piece, and here is another piece. So they cannot combine together because they need higher energy level. But if they sit here into the enzymes active site, this is the active site that matches with the substrate, that they can snuggling in into that active site. Sometimes the active site has some charges too. And in this case, the substrate can easily get in, like this will be positive, this is negative, so gets in here, induced fit model. And the enzyme is going to facilitate the reaction for that they would fuse together. So the output will be like something like this, okay? Does it make sense? So it will be positive and negative in here. Okay. So the other enzyme, let's say it is something like this. And its substrate, this cannot sit in here. So this will be enzyme B, and we have a square. For example, the square sits in the active site. This is the active site where the substrate sits, and this will split that in two. So it's an exergonic enzyme that facilitates exergonic reactions and makes 2 out of 1. So what I wanted to model in here for you guys is that the enzyme is specific for its substrate. The substrate will change, substrate product will change, but the enzyme does not change during or after the chemical reaction. It stays back and will facilitate more chemical reactions naturally. Okay, so it's reusable material, but very specific for its substrate. We cannot mix and match those. We have as many chemical reactions we have in our body that many different enzymes we have in our bodies. And those enzymes are made up of proteins. They have special shape, and the special shape can be distorted by or ruined by high temperature, as the high temperature will break the tertiary structures for sure. So the shape changes, the function changes. This is another main topic in biology, structure to function. If the structure changes, the function of the material will change too. And that is true for the enzymes too. Now, we talked about the enzymes. And before we move on, I would like to talk about a little bit more, like every enzymes is specific for its substrate, but also the enzymes perform their work in different environment, and they can be in their best shape by facilitating the chemical reaction with the highest rate in a certain chemical environment. Let's say we are talking about the stomach. Here the pH is low, let's say 2, and we have an enzyme in the stomach called the pepsin that breaks down protein in the stomach, and it works best at 2 pH. The y-axis is the rate of activity, and this is the pH in here. Now let's move into the mouth. In the mouth we have the amylase that starts to break down the starch. And let's say this is going to be like this. At 7, this is the amylase that works the best at pH 7. Out of its range, the rate of activity will be super low because the different pH affects the tertiary structure of the protein. Structure changes, function will change. And later on, they will not even do anything. The third enzyme is going to be the trypsin. Let's say this is in here. This is trypsin. This is another enzyme. This is, let's say, pH, and this is in the small intestine. The small intestine, the trypsin acts the best at alkaline pH when the pH is high. And this is how the rate of activity would look like for these 3 enzymes. Based on the pH, their rate activity changes too. Next topic. So we talked about the enzymes activities and the rate of activities. How are we going to figure out the rate? So it's rate calculation. It's kind of a skill for students to know on AP Biology. I tried to draw you guys a graph. And the y-axis will be the time. The unit will be in seconds. Let's say 1, 2, 3, 4, 5, 6, 7, 8, 9. So this is 1 second. 3 seconds and 5. Okay. The y-axis will be the product formation. We try to figure out, so if we measure the product formation, the unit will be for that in milligrams. How many grams of milligrams of product will be produced by that enzymatic reaction? We can figure out the rate of the enzymatic reaction. And also we can compare 2 different enzymes too. Let's see, this is 2, 4, 6, 8, 10. So 1 square will be 2, 6, 2, 4, 6, 8, 10. That's good. So I tried to plot the graph for you guys in here. And 1 and 4, 3 is going to be 10. And okay, so it will be something like this. Okay. Now, I would like to find the rate between 0 and 1 time. So rate for the enzymatic reaction between 0 and 1 second. Figuring it out is the rise. Okay, how many did it rise? 4 milligram rises over run. How many seconds was that? That was 1 second. So pretty much the rate is 4 milligrams per second. That's its rate. Can we figure out the rate for that enzyme, for the same enzyme between 1 and 3 seconds? How fast is the product formation at that time? What is the raise for 1 to 3? So here is going to be the raise. Now it was 4 and 10. 10 minus 4 is going to be at the top. That is pretty much 6 milligrams. And the run is up till to 1. So 3 minus 1 second. This is 2 seconds. And we have to do the division. For that, it's going to be 3 milligrams per second. So the rate is slower at this area compared to the beginning of the area. We will not figure out the rate in here after third second, because we see it's getting less and less the rate. I would say this is a slow rate. The slow rate is coming from that the enzyme probably used up all of the substrates, so no more substrates is in the system to produce product. Can be like that. This is the highest rate. The highest rate is here. Why is that? Because we have tons of substrates and tons of enzymes. So they are pretty much bind each other and they form the product. If I increase the temperature a little bit, just a bit, it will be even higher, the rate, because the particles are moving even faster and the collision of the substrate with the enzymes is more likely to happen up until a certain temperature, because after a certain temperature, the enzyme gets denatured, loses its three-dimensional shape because of the tertiary structures, hydrogen bonds, and interactions are going to be messed up. Its active site will get distorted, so it will not function anymore. What else in here? The rate depends on the substrate concentration, the enzyme amount of the enzyme concentrations, and the temperatures. My question, how would the graph look like if we increase the substrate level, but we would not change the amount of enzymes? If the enzyme is not the limiting factor, well, in this case, if I increase the substrate level, probably it will be like this. Okay, faster because we still have tons of enzymes and more substrate, we can form more products at that point. How would the graph look like if we increase amount of, what did we increase in here again? In here, if we increase, well, if we increase the substrate, it will be like increased substrate concentration. More product will form. If there's enough enzyme, it will be even a faster rate. If we increase, I think it's good. If the enzyme is the limiting factor, but we increase the substrate concentration, it can be like that too. Okay, same. It can be like that too. I go with that. Okay. Number 6, 0.6 at this point is, how are the enzymes controlled? Because we need to control the enzymes, or the body controls the enzymes. So controlling enzymes. So we have a substrate like this, the circle, and with the help of an enzyme, it can turn into a different substance, a product. We have another enzyme, enzyme A, enzyme B, will produce a different product. If we block this enzyme, so we block it, what would happen with the circle in that area? Its amount will increase and the square's amount will decrease. Therefore, the triangle will get decreased too. Okay, so the enzymes can be controlled this way, or sometimes the product, if we have too much. So this is one option. Sometimes the product is controlling the enzyme. So if we have tons of squares, this will block enzyme A. Hello, we don't need more squares. Stop converting the circle to square. How are they controlled really, these enzymes? We have two types of control: competitive inhibition. This is one of them. When we have an enzyme, we have this enzyme. And let's say this enzyme is cutting the triangle in half. It will sit in here. And when it comes out, it will be 2 triangles. And in our body, there will be another similar shape substrate like this. So this can sit in here and blocks the enzyme's activity to convert the triangle to 2, but instead this one is sitting in here. This is a competitive inhibition because this shape, this material competes for the active site of the enzyme. This inhibition depends on the substrate concentration. I can overcome this competitive inhibition by increasing the triangles. I will have only 5 diamond shape, but 100 or 1,000 of triangles. Well, in this case, the competitive inhibitor will not be as effective. But on the other hand, we have another inhibition, and it's called the allosteric / non-competitive inhibition. At this point, the enzyme, we can just do a different enzyme. Kind of the active site is like this. And the inhibitor will be something like that, sitting at the allosteric site, combines with the allosteric site. And that allosteric site will change the shape of the active site. So no matter what, I increase the substrate material, the squares, this will stay in here and it will be this type of inhibition is independent from the substrate level. No matter how much I increase the squares, it will not increase the rate of reaction. And the last thing what we have to talk about is the experimental controls. Experimental control, in an experiment, we have the controlled group and we have the experimental group. Control group is usually a standard measurement to compare our experimental control group's result. The control group can be divided into negative control and positive control. The negative control is something that it's not a group of specimens, that they are not exposed to any kind of material. And this is the group, the negative group, that confirms that the result is what it is without any treatment. So let's say we want to just measure checking the reaction rate of an enzyme with a chemical, and the negative control will be everything in the test tube but the enzyme. In this case, we will see that nothing will happen to the substrate in the test tube without the enzyme. We exclude that the temperature, the sunlight, or anything else would affect the results. We will see that the result is slowly only depended on the presence of an enzyme. So that's a negative control. No manipulation to the group, the negative control group. On the other hand, the positive control group is a group of specimens that they get the treatment with a known effect. Like, I want to test the different light sources for the plants. In this case, the experimental group would be the plants with red lights, plants with green lights, plants with blue light, and their growth, measuring the rate of photosynthesis, for example. And the positive control will be when I have the plant with normal light outside in my garden or in a controlled environment, but a light that contains all of the light spectrum, basically. So that is the positive control. We will learn about it a little bit more as the time goes on, but I wanted you guys to know a bit about them. And that's it for today, for the office hour. It became a little bit long, but please let me know if you have any questions. Have a good one. Bye.